All right, and for today, we're going to be going over chapter 24. We're going to talk a little bit about the assignment and what you're going to do in terms of scheduling for your presentation at the very end of the semester. And we'll have a couple lab assignments. And then we'll talk a little bit about the structure of your paper as well. Okay. So we'll go ahead and start on chapter 24. All right, an overview of the immune system. So what is the immune system? So the immune system is a body system of defenses against infectious disease, right? And this is very important. And it's kind of been a topic that's been recently brought up in the news about individuals that are susceptible versus individuals that are not as affected by a disease, right? Especially when we're talking about COVID-19. A lot of people are in a category that are immune compromised versus not immune compromised. And what's the difference, right? And essentially what we're referring to is the body's ability to fight off infection, okay, from a pathogen, right? And a pathogen is just a disease causing a virus or organism. So it can be a bacteria, virus, fungus, protist. There's a lot of different organisms that can cause infections, right? And we have two types of immunity that make up our immune system. We have the innate immunity, which is the part of the immune system's defenses that is always present in its final form and ready to act. And these are going to be your cells, right? These are going to be those cells, those white blood cells that are floating around in your blood and your body that will make contact with a pathogen and also fight them off. There's also something in the innate immunity called the complement system, which for this class we won't get into too much, but just know that it's always there, and it's pretty much just proteins that are found in your body that can react to pathogens present in your body. Okay. The innate immunity is very important for fighting off fungal, bacterial, also viral infections, right? Your cell-mediated responses are important for that. Okay, your adaptive immunity, which is very important for vertebrates, only found in vertebrates. And so you don't see this in invertebrates, so those like porifera or the sponges, and then so it's early evolved organisms. The adaptive immune system is a part of the immune system of defenses that is found only in vertebrates and that must be activated by exposure to a specific invader, right? So this is going to be referring to your antibodies, okay? Your learned responses. So a part of the immunity that makes the antibodies, your B cells that eventually turn into your plasma cells that release the antibodies, your immunoglobulins into your bloodstream, okay? All right, so the lymphatic system. So this is the organ system which lymph circulates, includes lymph vessels, lymph nodes, and several other organs. The lymphatic system helps remove toxins and pathogens from the blood and intersagittal fluid and returns fluid and solutes from the intersagittal fluid to the circulatory system. So this system works very closely with your circulatory system, and you have lymph vessels surrounding your body, especially in points of aggregation of your blood vessels. So on the inside of your groin, your armpits, on the sides of your neck, you have lymph nodes, right? Sometimes what you'll see is you'll have individuals who develop cancers in their lymph nodes. And this is quite common because, again, this is going to be filtering out anything that's present in the blood that shouldn't be there, right? And sometimes that's going to be cancer. So typically what you'll have is breast cancer, and then you'll have metastasized cancer cells that go to the armpit where you do have a lymphatic vessel there, the drainage vessel there. So you'll have cancer that leaves and gets stuck in the lymph duct that's in your armpit. All right. Same thing with your groin or your neck, right? If you develop an infection, you have some sort of cancer, you have the metastasization into your groin or your neck, right? You have the swollen neck, or you'll have a throat infection and you'll have the lymphatic duct that would be swollen. Like I recently had strep throat and both my lymph nodes were completely swollen. Okay. And again, it's just because that pathogen or that reaction that's going on with the microorganism that was present in my throat is being accumulated in these lymph nodes that are found in my neck, right? So it's filtering out everything that can potentially be bad. All right, so here's the innate immunity. The innate immunity includes skin, secretions, mucous membranes, including your stomach acid, right? This helps protect against infection or invasion of your GI tract. Your skin or the integument system is also incredibly important for protecting against infection. This is all a part of the innate defenses, right? You have IgA antibody that's secreted in your saliva as well as your tears. You have different enzymes that are present that break down bacterial cell walls that are present in your saliva and in your tears. You have phagocytic cells. This includes basophils, eosinophils, neutrophils, monocytes, activated monocytes. Right? These are going to be those cells that are floating around in your blood that are ready to attack or ingest a pathogen. Those are natural killer cells, defense proteins, inflammatory responses. Okay, so these are all very important for fighting off any potential infection. We'll talk about those in a minute. All right. So the adaptive immunity, right? These are internal adaptive defenses. You have lymphocytes. So the cells that mediate the adaptive immunity are the B cells and the T cells. And one of the major components of the adaptive immunity is actually these antibody responses, or this antibody, these proteins that are secreted by the B cells. Okay, well, they're actually secreted by plasma cells. So what happens is a B cell will be activated by a T cell typically, so it'll come and touch the B cell with antigen, and this guy here will go okay, well, it's time to make antibodies against whatever the T cell had shown the B cell, and it'll become activated. Then this B cell will typically migrate from a lymph node, so either your groin, armpit, neck, right? It'll migrate from there to your bone marrow, where it'll start producing and secreting antibodies into the blood, okay? Which are these guys here, these Y-shaped little things. And we'll talk more about that in a minute, okay? And the lymphatic system, this is involved in internal innate immunity and adaptive immunity, right? So both innate and adaptive. And these forms of immunity work very closely with each other, right? The adaptive immunity requires the innate immunity in order to help modulate it, right? And the adaptive immunity is incredibly important to the innate immunity because it provides another level of protection, and it also provides memory. Okay, so you do have memory B cells. This is why we're able to vaccinate individuals, and you can still produce an immune response and protect them against any pathogen that can cause a severe infection, right? And we'll get to that in a minute. All right, so external or innate defenses block out or filter out pathogens. All right, skin forms a protective barrier. The outermost layer of skin is brown, consists of dead cells that are continually shed and replaced by layers of living cells below. Okay, so you see the dead cells here, and then you see that epidermal tissue growing outward and dying eventually. And this provides a nice layer um, that protects the body from any pathogen that's trying to invade. Here you have cilia on the nasal cavity. So you have mucus in blue, traps bacteria, which are in yellow, and sweeps them out of the body with hair like cilia, right? So those of you that are moving on to anatomy and physiology will get to learn about pseudostratified columnar epithelium. And again, these are this epithelium tissue that's lining the upper respiratory tract as well as your lungs and your bronchus. And these cells essentially push mucus out of the lung up into the upper respiratory tract, and then either the mucus can be spit out or swallowed. Okay, and this constant process is very important. Excuse me. This process is incredibly important because moving the mucus up and out of that lung, the lung tissue, allows for the lung to be incredibly clean or almost near sterile, right? And you want this. is if you allow bacteria to accumulate and stay in the lung, it's wet, warm, there's plenty of food there. You can have issues for those individuals, right? There's actually a disease called cystic fibrosis that prevents the mucus from being allowed to be removed from this mucociliary escalator, is what we call it, because it's just essentially it's an escalator that lifts up the mucus up and out. And these individuals with cystic fibrosis suffer from serious infections that people with normal functioning mucus um, don't get. Okay, and those individuals don't live very long. Right? It's one of those rare genetic diseases that actually very very severe for that individual that has it. They don't. The life expectancy is incredibly shortened due to the disease. Okay, um, but normal functioning cilia, again, this is a part of the innate immunity, just catches or captures the bacteria with a sticky sugar protein and then lifts the bacteria out, and then you can swallow or spit out the mucus, whatever you would like to do. There's some conflicting evidence. I've heard people say don't swallow, and I've heard other people say do swallow, but the mucus when it comes into your mouth or upper respiratory tract, we're made to swallow it. It's almost natural, right? It almost pops up, and you never even have to get it into your mouth, and the mucus can fall back into your GI tract. Okay, um, yeah, I've heard a few different things though from actual professors that are telling us do or don't, but I I'm pretty sure it's from what I've read, your body's built to cough up the mucus, and then you're built to swallow again that lower pH. Also, we have something in our guts called MALT, or mucoid associated lymphatic tissue, and it's essentially part of the lymph system. And what that tissue does is it actually samples our whatever we're eating, and it will decide whether or not we're eating proper things or whether or not the bacteria in our gut is okay to have there. Okay. So that mold can give us a warning of potential pathogens. Given that we're swallowing the mucus and it's capturing any microorganism that's coming into our lungs, it can tell us whether or not we should be worried about any pathogen that's on our tissues. Okay. But we won't get into that. It's a little more immunology than anything, advanced immunology. All right. So internal innate defenses, we have a phagocytic cell, a white blood cell that engulfs bacteria, form proteins, and remains of dead body cells as part of their internal innate defense. Excuse me. You have natural killer cells. This is a white blood cell that attacks cancer cells and infected body cells as part of an internal innate defenses. An infected body cell typically refers to a cell that's infected with a virus. Okay, and that virus will cause something called cytopathic effects, and this just shows a different type of cell rather than a normal cell, right? So think of a cell that's just sick and doing things that aren't appropriate, right? These natural killer cells can recognize that, and they will go and they'll touch and trigger that cell to perform apoptosis. What that means, apoptosis, is controlled cell death. The cell will just say, all right, well, it's time to go. Thank you for letting me know I need to die. And the cell dies. And essentially it's taking with it any infection or any virus that's inside of that cell. Okay. And we'll get into that in a little bit. But phagocytic cells are those cells that typically make a pus. They're floating around your body. They'll go and they'll eat things and make sure they're not going to be causing problems for you later. Inflammatory response, this is an example of internal innate defense involving in release of histamine and other chemical alarm signals, which trigger increased blood flow, a local increase in white blood cells, and fluid leakage from the blood. The results include redness, heat, swelling of the infected tissue. Okay. Histamine, this is a chemical alarm signal released by injured cells that cause blood vessels to dilate during an inflammatory response. Okay. So histamine, again, very, very strong. We take medications called antihistamines, right? Antihistamines essentially stop that inflammation of the upper respiratory tract when we do have an allergic reaction, okay, or allergies, right? And this is going to be more severe because those cells that mediate that, so our mast cells and other immune cells, that response is going to be very strong. Okay, so it takes a little bit of a reaction of those cells to release the histamine in the upper respiratory tract to cause those severe reactions, the leakage, the runny nose, watery eyes, and heat or swollen upper respiratory tract. Okay, inflammatory response is a little more important to understand. Inflammation is important, right? And everyone always talks about inflammation being bad. You don't want inflammation. And I'm here to tell you that no, you do need inflammation in certain situations, okay? Maybe not if you blow out your knee or roll your ankle. You don't want to recruit immune cells, but eventually you do. Eventually, uh, um, warmth and inflammation is good in that body site, okay? Um, especially because inflammation means that there's going to be reconstruction of tissues. There's going to be involvement of the immune cells, or inflammation, the inflammatory process. Okay, and you need that, right? So if you, I don't know if any of you have ever gotten burned, and it seems like maybe an hour or 30 minutes later, it gets really hot, and it gets more sensitive than when it was after you initially burned it, right? So after, if you burn yourself severe enough, you have the blisters or this swelling. It almost hurts more 30 minutes to an hour later, and it's because of that inflammatory response, right? Your white cells are being recruited to that area. Um, your body is starting to repair itself. It's starting to say, "Hey, all these cells are dead. We're going to kill these cells off, and we're going to start growing or releasing growth factor and different chemokines and cytokines in order to recruit white cells to the area to prevent infection. And then we're also going to start growing." Okay, and that can be painful, right? You're gonna have all this innervation of different immune cells, and you're gonna be expanding or growing that tissue back. Okay, and same thing with the cut, right? It almost seems like you cut yourself initially. It doesn't really hurt, even if it's a deep cut, and then within an hour or so, you start feeling the hot and the pain. And again, that's from inflammation. That's good. You want that there, right? You don't want to cut yourself and nothing happens, right? Inflammation is good in terms of reconstructing tissues and protecting your tissues from an infection. Okay, so you do want inflammation, right? All right, so here's a nice little shows a couple of steps. This figure shows the steps of inflammation, right? So whenever tissue is damaged, the body responds with a coordinated set of internal innate defenses called the inflammatory response. Okay. So here we have the skin surface, that dead layer, and then here we have the penetration of a splinter, right? So the splinter has introduced bacteria. Now we have the chemical signals released by the cells to recruit to recruit white cells or other reconstructive white cells to get rid of this. Okay. So tissue injury and release of chemical signals such as histamine, cytokines, chemokines. These proteins that are released by these cells are going to be calling for white cells to move out of the blood and into the tissues. Okay. Oops. So this movement of white cells to the tissues is called extravasation or diapedesis. Okay, so extravasation or diapedesis. I've heard it in two ways. I've heard an endocrinologist call this extravasation, and then I've heard an pathologist call this diapedesis, and I got used to calling it diapedesis. Okay, so this is just the movement of white cells out of the blood vessel into the tissues in order to start that process of reconstruction and helping that heal, that area heal. Okay, so if you didn't notice, these immune cells will start dumping digestive juices, and they'll start consuming these bacteria. They can even dump digestive juices that'll break down this splinter. Okay, um, and you'll see the white cells coming in and kind of helping that infection, right? To make sure that bacteria doesn't spread into the blood and you don't go bacteremic, right? Because you don't want this to happen, right? You don't want the bacteria to come and get into the bloodstream and cause infection systemically. So here you have the phagocytes consuming or phagocytizing the bacteria. Now you have the wound being healed and cleaned up, and you'll have less swelling and infection as the skin heals, right? So eventually it doesn't hurt as much anymore. Upon the initial cut or scrape, it typically hurts a lot. Okay, so um, yeah, so yeah, you remove, boom, you have the cut, and then the inflammation, the recruitment, or the diapedesis or extravasation of those white cells, and then eventually reconstruct that tissue and remove the bacteria. Okay, so remember, inflammation is important. So the lymphatic system. So you have a lymph node. This is a small organ that is located along the lymph vessel and that filters lymph. So lymph is a fluid similar to intrasagittal fluid that circulates in the lymphatic system. Okay. To provide the best protection from pathogens, the body's immune system relies heavily on interactions of innate and adaptive immunities within the lymphatic system. Okay. Um, so again, this is going to be very important for filtering out, for identifying pathogens, for ensuring that your body is safe and that you don't have any type of pathogen that's going to be invading your body in different areas. Okay, and so this is why you have multiple lymph ducts. Okay, also this can be a source of disease. So right, we talked about cancer spreading or metastasizing to the lymph node. Think of breast cancer, and then that lymph node closest to that breast will start having the swelling or the tumor forming there. You can also have filarial worm disease that causes lymph nodes to swell. So one of the main things that we've seen with lymph node or lymph diseases that's quite common and popular is elephantiasis. So this is the disease where individuals have their distal appendages or other parts of their bodies swell with fluid. And this could be from the plugging of the lymph node, right? This lymph fluid has nowhere to drain. And because of the filarial worm disease or the infection, the worms plug the lymphatic vessels and you have the swelling or the fluid buildup in the lymph node. Um, so here you have these lymphatic systems or lymph vessels. Remember, they're very closely related to the blood vessels, or they're closely aligned with the blood vessels. So in between the capillary beds, you have the drainage sites of the fluid that goes into the lymphatic vessel, right? And in the lymph vessel, you have a bunch of immune cells that are hanging out. You have T cells, B cells, you have phagocytes, you have all different types of immune cells that are hanging out in these vessels that are ready to go. And essentially they're waiting to be activated. They're waiting to touch something that's bad, right? They're just hanging out with their receptors, waiting for a pathogen to bump into them so they can stick and then grab and be like, hey, this is potentially bad, right? So the primary lymph vessels that we have are the spleen and the thymus and bone marrow. We have secondary lymph organs. I'm sorry, I think the spleen is a secondary lymph organ. And then you have all of these other lymph vessels, these little nodules that you see. And again, um, you'll have them everywhere, right? You have them in your neck, you can feel it, your armpit, your groin, and sometimes you'll feel the swelling of those lymphatic vessels too. Okay. All right. You know what? Let me show you that. So this is elephantiasis. So this is a filarial worm disease that I was talking about. Um, and essentially, you have this little worm here that can plug your lymphatic vessels, right? So here we have the it's called elephantiasis, but it's caused by a filarial worm. Okay, and what this filarial worm does is it becomes injected into the human, um, and upon the injection in the human, you have the worm will come and it'll have a few different life stages, but it'll plug a lymph duct. Okay, and this lymph duct then will swell with fluid, and then you can have severe swelling of the distal appendages, and it's typically in impoverished areas, right? Because if this starts occurring here, we have subtle swelling here. We would go into the physician right away to ensure that this gets taken care of, right? We can get treated for that filarial worm disease, therefore preventing the swelling of the distal appendages, right? It's a terrible disease. And again, this is because this worm gets plugged into the lymph duct and the lymph vessel, and then you can't do anything about it. So think of heartworm in dogs, right? Heartworm again will get into the heart of the dog and cause issues with pumping and blood flow, right? This essentially is the same thing, but instead of getting into the heart of humans, it gets into the lymph duct and it causes this swelling. Okay, and after this tissue has been stretched out, there's really nothing you can do to fix it. Um, it's quite sad. Okay, so. Um, yeah, you didn't know about filarial worm disease. Now you do. Elephantiasis. Okay. But again, it's a disease of the lymph duct. It's from the plugging of this lymph duct and then the swelling of the appendage. All right, so lymphocyte. So this is a type of white blood cell that carries out adaptive defenses, recognizing and responding to specific invading pathogens. There are two types of lymphocytes, B cells and T cells. Okay. All right, so adaptive immunity, B cell. This is a type of lymphocyte that matures in the bone marrow and later produces antibodies, okay, to help combat pathogens circulating in the blood fluid, okay? So this B cell will hang out in a lymph node, let's say the armpit, groin, neck, right? And then eventually, if it comes into contact with a pathogen, it becomes activated, right? And then that's when it will go into the bone marrow and then start producing the antibodies, okay? And these are the antibodies that we associate with the adaptive immunity. Okay, T cells. This is a type of lymphocyte that matures in the thymus. So the thymus is an organ found in the center of your chest. And again, it's required. It's a primary lymph organ. So antigen is any molecule that elicits a response from a lymphocyte. Okay, so it's anything like a sugar or a protein that's found in a bacterium or a fungi, to pieces of a virus that are broken down in the bloodstream, right? So there's a few different things that can activate or that can be antigen, right? And that cause these cells to react to it. Okay. So two ways antigens are recognized by adaptive defenses. Note the B cells and T cells bind to the antigen, but T cells additionally require that the antigen be bound to a self protein. Okay. So what's going on here is antigen recognizing by a B cell. So you can have activation of B cells, but you have a much stronger response, okay? Here, for if a T cell activates a B cell. So this is actually a T cell that's sampling our own cells. And this is important, right? So there's different types of T cells. There's natural killer cells, there's T helper, so there's CD8 cells, and there's CD4 positive T cells, okay? So there's a lot of different types of T cells. Here is showing the natural killer cell or the T cell that's going to be sampling our own body cells, okay? Oh, this might be a little confusing to explain, but I'll explain it anyways. Okay, so B cells are important for making antibodies, right? They get touched by antigen, they go into the bone marrow and start making antibodies, right? Simple. It's just going to be making antibodies against pathogens, okay, which is important. You can also have inappropriate responses, but we'll talk about that later, okay? T cells have a few different functions, right? You have CD4 positive T cells, CD8 cells, T killer cells, or natural killer cells, or cytotoxic T cells. So the CD4 positive T cells are those CD4 cells are going to be responsible for activating or help activate B cells. Okay, so those T cells are not going to sample body cells essentially, right? They do, but they won't, right? They're not that important for that. The cytotoxic T cells are those T cells that are sample the body cells, essentially are looking for cancers and looking for viruses. They're looking for cancer cells that need to be destroyed or killed, and they're looking for cells that are infected with viruses. And you have these little receptors. They're called MHC1 complexes, whereas T cells have MHC2 complexes. All immune cells have MHC2, but these body cells are sticking out antigen from themselves and saying, "Hey, I am you. This is your body. don't kill me. Okay. If this body cell doesn't display that, then there's an issue. That means the cell's either cancerous, it's infected with the virus, or it's something's wrong with that cell and it became faulty. Okay, so it needs to be displaying self protein in order to work properly, in order to be okay. Okay. All right, and that T cell needs to recognize that. If it doesn't, this T cell will come, this natural killer cell will come and say, well, it's time for you to die. And it'll touch and cause something called apoptosis, which means it's just a controlled cell death, and that cell will die. Okay. Uh, so, yeah. Any questions about that? If anyone has any questions, please let me know. Those of you who are watching the recording, if you want me to explain this in more depth for where you can understand, by all means, come to office hours or bug me. I'll be more than happy to explain this process in more depth. Okay. All right. So step one is the activation. So the recognizing the invaders, the activation. Okay. Step two, cloning the responders. Clonal selection. So this is a production of a population of genetically identical lymphocytes, white blood cells that recognize and attack a specific antigen, right? That stimulates their proliferation. So it means it stimulates their growing. Okay. Clonal selection is a mechanism that underlines the immune system's specificity and memory of antigens, right? So clonal selection just means that there's going to be a B cell that's activated by the antigen. It's going to eventually turn into a plasma cell that produces the antibodies, right? And you cannot just have one cell. You need many cells that produce multiple antibodies at one time that are going to target one specific thing, right? So this is why it takes a little while for the adaptive immunity to function properly, right? This is why you will get sick with something and it takes a week or a few days for you to start feeling better if you need to be treated, right? This does take a little while to function properly, right? Your innate immunity will help mediate or modulate that infection, but your adaptive immunity is going to help protect you against the infection later on. Okay, and you'll have memory there. So you have an effector cell. This is a short-lived lymphocyte that has an immediate effect against a specific pathogen. You have an antibody, which is a protein that is secreted in a B cell and attaches to a specific kind of antigen, helping counter its effects. Okay. So essentially, an antibody is made up of protein that just binds to other organisms or pathogens or antigens. Okay. So it can even be a virus. It'll bind to the virus and essentially stop that virus from working properly. Okay. So clonal selection during the first exposure to an antigen. In this example of clonal selection, an antigen binds to a B cell and causes the cell's proliferation. A similar kind of clonal selection operates on T cells. Okay, so antigens bind a B cell with complementary receptors. So this antigen can be pieces of a pathogen, virus, fungi, bacterium. Antigen receptor on cell surface. Again, it'll activate this. Um, Binding causes the B cell to divide, forming a clone of identical cells with the same antigen receptor. So upon activation, it'll induce the cell to replicate, right? Some cells develop into short-lived effector cells that secrete antibodies specific for antigen. So this cell's job is just to dump antibody out into the immune system or out into the systemically into the body, right? So this takes a little while to divide. It takes a week or so to get this fully ramped up, right? So these cells are going to be essentially dumping the antibody to protect or antibodies to protect your body from the pathogen, right? So it's going to be just continually dumping out, and eventually these cells will die. Okay, so some of the cells develop into long-lived memory cells. If later exposed to the same antigen, they produce larger clones of membrane effector cells, right? So these clone memory B cells are going to be housed in your bone marrow. And then if that antigen comes back, let's say you get another infection with the same organism or the same virus, this provides a quick level of protection, right? This allows for your body to respond more rapidly to that infection and more appropriately. Okay. So this is why we utilize vaccines. okay? In order to have these memory cells that if one day you come into contact with that pathogen again, or you come in contact with a pathogen you're getting vaccinated with, you're gonna have a much quicker response time, right? It's gonna be much faster. You're gonna be safer if you have been exposed to the antigen beforehand because it provides a level of protection, okay? And it provides a level of faster protection. That means you have antibodies secreted right away, um, rather than taking a couple of weeks to develop the memory or clone B cells, right? That by then the pathogen could have ran through your body and you might be dead. Okay. All right. So primary immune response, the initial immune response to an antigen, which includes production of effector cells that respond to the antigen within a few days, and memory cells that will respond to future exposure to the antigen. So a memory cell is a long-lived lymphocyte that responds to subsequent exposures to the specific pathogen. A memory cells form during the primary immune response and is activated by exposure to the same antigen and that trigger this formation. When activated, a memory cell forms large clones of the effector cells and memory cells that mount the secondary immune response. So secondary immune response is the immune response elicited by memory cells upon exposure to a previously encountered antigen. The secondary immune response is more rapid, of greater magnitude, and of longer duration than the primary immune response, right? Secondary immune responses are always better. And this is again why we have vaccinations, okay? All right. So responding to invaders, we have T helper cells. This is a type of lymphocyte that helps activate cytotoxic T cells to attack infected body cells and helps stimulate B cells to produce antibodies, right? So we have CD8 positive T cells that help prevent against cells that are infected with viruses or with cancer. And then we have CD4 positive T cells. These CD4 positive T cells help B cells become activated, right? So I know you've seen this B cell be activated by antigen, but I'm here to tell you that this is not good activation. This actually doesn't produce memory cells. Okay, the book kind of got it wrong. Sorry, book. But unless it's a very strong response. But typically, what happens is a T cell will come, the T helper cell, CD4 positive T cell will come and bring antigen. So let's say there's a T cell here, it'll come and bring antigen here and touch the B cell with the antigen. So it'll antigen present. So it's called presenting. So it's like if I were to go to my boss and say, hey, there's a problem. At the work site. Let's say it was at the hospital. Hey, there's a problem here. I need you to fix it. Okay. And this is the boss. The B cell's the boss, and the B cell's going to be like, all right, well, I'm going to get together and I'm going to make a bunch of things happen, and then I'm going to dump the solution out for you to fix whatever the issue is, right? And this is essentially what happens here. The T cell is going to be the worker that's going to go to the boss to get things done. Okay, and that B cell will eventually get those things done that are required. But the T cell, CD4 positive T cell activation is an incredibly important part in the immune response. It's incredibly important. This provides a level of activation that's not normally seen, right? If this antigen just touches a B cell and then you have the activation and you have the secretion of antibodies, this is not as a strong response as, let's say, if a T helper cell comes and touches this B cell. If the T helper cell comes and activates that cell, this is a much more potent response. Okay, so these cells are incredibly important for the immune system, right? Enough so to where HIV actually targets the CD4 positive T cell, and we've seen the CD4 positive T cell in humans become depleted, especially with individuals with HIV, and those individuals suffer immensely, right? So individuals that do have HIV/AIDS actually don't die from HIV/AIDS. They die from symptoms or pathologies associated with HIV/AIDS. Okay, so the virus does infect these CD4 positive T cells, but the CD4 positive T cells being gone don't cause the individual to die. The absence of the CD4 positive T cells means you lack a very crucial component of the immune system, and it opens the body up to new types of infections, right? So back early on, when HIV had not been discovered yet in population, they started noticing trends. Pneumonia that shouldn't be in individuals as young as those who were developing it. They saw different types of cancer, Rousey sarcoma, um, which is caused by a herpes virus that eventually leads to cancers, right? And this was almost unheard of, the developing overgrowth of HPV. So some of these individuals would develop immense amounts of warts that were uncontrollably growing, and they didn't know what was going on. Well, if you remove a very important component of the immune system, such as a CD4 positive T cell, you lack the ability to fight off these viruses or these pathogens or these bacteria, these bacterial fungal organisms that are causing these infections. So the individual would pass away from these various infections that typically didn't happen in normally healthy adults. Okay, and then they realized, oh shoot, um, there's a virus that's been passed around for a few years now, and there's a lot of people that are infected, and there's a lot of people that are dying. Okay, all right. So the roles of activated T helper cells. So T helper cells stimulate both arms of the adaptive immunity defensive. So cytotoxic T cells and B cells. So um, this is antigen presenting. So again, this is where the phagocyte will come and say, hey, look at what I got. Please take this to your superiors. This T helper cell will then become activated, and you have clonal selection. So you have this occurring at the same time. So it's copying bits and pieces of itself because again, it recognizes the antigen. Therefore, it's going to make more copies, and then you'll have more of this type of T cell that can recognize the antigen, right? And this will cause the activated T cell will activate other cells, either the cytotoxic T cell or the B cells. So cytotoxic T cell will attack infected cells such as viruses or cancers, um, and then the B cell will dump antibodies out once it turns into a plasma cell. Okay. So the effector B cells secrete antibodies and bind antigen. All right, so these antibodies bound to antigens on the surface of pathogens, right? So here you have the antibody again binding to the antigen. This can essentially neutralize an antigen or neutralize a pathogen once it's bound, right? If multiple antibodies can bind to a bacterium, that bacterium won't do anything. Same thing with a virus. If antibodies can bind to that virus, the virus will then be deactivated or will not have functioning anymore, okay? Essentially like covering something with glue and preventing it from leaving or causing issues. So here's the antibody binding to a virus. This virus is trying to gain entry or access into the cell, but excuse me, if the antibodies bind to that specific virus, it essentially neutralizes it. Also, these antibodies that are bound will cause something called opsonization. And opsonization, what I like to think of it as, is making this tasty. So making bacteria tasty. It's almost like salting your food, right? So think of your body releasing the antibodies and sticking to these bacterium or these bacteria. It's making it tasty for the phagocyte to come and recognize this, like, oh, nice. There's these FC regions here on antibodies. So these FC regions are attract phagocytes, and these phagocytes will then be more likely to come and digest or break down the antibody antigen complex shown here. Okay, this clump. So cytotoxic T cell response is the sole function of billions of your cells. The sole function of billions of your cells is to kill other cells. So cytotoxic T cell is a type of lymphocyte that directly attacks body cells infected by pathogens. Okay, and this can be true in terms of if it's infected with a virus, this cell will come and kill your cell because it's infected with that virus. There's really nothing you can do after that. But this cell is also important for stopping overgrowth of cancers, right? If you have a cancer cell that forms, this cell can come and say, hey, you look suspicious, you look kind of weird, I'm going to kill you. Sorry. And it'll just come and kill that cell. And that's important, right? You don't want overgrowth of cancer. You don't want cancers to metastasize. This is this cell is a very good mechanism of preventing overgrowth of anything. Cells, viruses or virally infected cells, right? So it's important to have this cytotoxic T cell response, because otherwise you'll develop severe viral infections or you'll develop cancers, okay, that you should normally be able to take care of. All right, so here you have the activated cytotoxic T cells. So cytotoxic T cell binds to the infected cell, becoming activated. Boom, you have the antigen, right? So you have the proteins again on this cell. Proteins that trigger cell death enter the infected cell, and then you have apoptosis. So the cell dies. But this is a controlled cell death. So these proteins will then be dumped into this cell, and it tells the cell, hey, it's time for you to die. Please kill yourself and fold yourself up nice and neat, so you don't dump all your digestive enzymes out into the tissues or your contents, right? Because imagine you don't want this to pop and dump virus out into the environment. You want this to be internally digested and this be nice and folded up and easy to package or move. Okay. All right. So memory cells, again, remembering the invaders. So step 4 allows for quick response to reinfection. Vaccine, this is a harmless version or piece of pathogen, a disease-causing virus or organism used to stimulate a host's immune system to mount a long-term defense against the pathogen. All right, so I wanted to talk a little bit about vaccines. They're incredibly important, right? So think of smallpox or polio, which essentially we've almost wiped off the face of the earth because of the vaccines that were created for it. This process takes a while, okay? So in order to create a vaccine, we need to find a protein or some sort of antigen that binds to B cells or binds to T cells that activates the immune system appropriately, right? We want something that can elicit an immune response. And this is why a vaccine can sometimes cause fever or headache or chills, right? Because it's telling your body, hey, you're infected. Please mount an immune response. The best immune response that you get is actually from an infection, though. So sometimes it's really difficult to create a vaccine that works appropriately, right? Because you could be injecting random proteins from a virus, and you may or may not have an appropriate immune response to those proteins. The best types of vaccines, excuse me, the best types of vaccines typically utilize what is called a attenuated virus, okay, and or bits and pieces of a bacterium that cause a very strong immune response. And what an attenuated virus actually is it actually causes a mild infection in that individual. So think of smallpox and cowpox. This is where vaccines were discovered. The advent of vaccines were discovered because milk maidens that would come in contact with cowpox regularly did not develop smallpox, because they would have these infections caused by a very similar virus to smallpox called cowpox. Okay. But they had an active infection. They would still get sick from the cowpox, but it was such a good immune response and cross reactivity with the antibodies, they never got smallpox. And the same is true here, right? Vaccines that are made with attenuated viruses are just much more potent, right? They activate the immune system much better, okay? Early on, there was a vaccine for polio that had an attenuated virus, so a weakened viral strain, but there's high risk, or not really high risk, but there is a risk associated with that attenuated virus. So some individuals who were immune compromised that took the vaccine, which they really, early on, this was an early vaccine. So certain individuals would be vaccinated by this attenuated strain, and some people were getting sick with polio from this attenuated strain, right? So they kind of pushed that one off to the side, because some individuals would get sick worse than what they were supposed to. They would actually develop the polio symptoms. But antigen-based vaccines are still very important. They still provide a level of protection, but the vaccines that actually utilize attenuated strains or attenuated viruses have a better or a nicer immune response, right? So here we have first exposure to antigens can be from a vaccine or no vaccine, right? You have the initial infection, the primary response, you have the antibodies being produced, the second exposure to antigen, and then you have an immense amount of antibodies produced in a much quicker time, right? So it's almost instantaneous, right? That you develop, you start producing and start cloning those memory cells in order to produce the antibodies. Okay. So immune disorders, we have allergies, which is an exaggerated sensitivity to antigen. Symptoms are triggered by histamines released from mast cells. So remember, mast cells are going to cause those allergic reactions, or you have the runny nose and the watery eyes. Okay. An allergen is an otherwise harmless antigen that causes an allergic reaction. Okay. So we have a Lone Star tick. So the saliva of this tick actually causes an inappropriate immune response or cross reactivity. So this tick will come and bite an individual, and the proteins in that tick's saliva cause your immune system to react to it, right? And this tick has very specific proteins that mimic a sugar, or sorry, they have very specific sugars that mimic a sugar that's only found in beef and red meat. So it's a sugar. Sorry, it's not a protein. I keep forgetting that it's a sugar. So the sugar is found in beef and red meat, um, and it actually causes an allergy to meat. So you actually will develop the tightening of the neck, the reaction, and the inappropriate response to meat. So you'll consume meat, you'll have like an allergic reaction like you would with peanuts. It's not as severe, but yeah, individuals can't eat meat for a long period of time. So I believe it's a few years that they're unable to consume beef or red meat after being bit with this tick, and you develop that initial immune response. And it's again essentially you're waiting for those cells in your body to stop being there. You're waiting for those memory cells to kind of taper off and you not have them anymore. All right. So the first exposure to an allergen causes sensitization. Subsequent exposure to the same allergen produce allergy symptoms. So you have pollen, which if you remember is a gymnosperms dumping their gametes out into the air. So allergen pollen grain enters the bloodstream, right? B cells make antibodies. Antibodies attach to mast cells. Mast cells release allergenic components into the body. So these histamines, very little needs to be produced in order to cause these allergic reactions, right? So the inappropriate immune response. So you're reacting to the pollen grains. Essentially, your body's freaking out when it sees it. Even though this isn't a pathogen, we're still treating it as a pathogen because it ended up in our body. Okay. So this is epinephrine, also called adrenaline. This injection can quickly stop life-threatening allergic reactions. So allergies have been kind of a big research topic in medicine lately because there's been so many different types of allergies, right? We have gluten allergies, we have milk allergies, we have peanut and tree nut allergies, right? There's a lot of different types of allergies out there. Egg allergies, so allergies to the albumin, right? The proteins that are found in egg. So this is a kind of a new big field of study in medicine because allergies have almost been identified as a newer a Western disease. Okay, and the research that I've looked at, looked at is kind of they don't know what to blame. They don't know where to point the finger, but there have been an immense amount of new types of allergies coming and in much higher rates, right? Where it's kind of been unheard of, right? Celiac disease or allergies to gluten, severe allergies to gluten. I'm not talking about people avoiding gluten altogether because they don't think it's healthy for them or good for them. I'm talking about people that get exposed or eat a small amount of gluten and they have bloody bowel movements within a few hours. Okay. So and severe cramping and the inability to eat food for the rest of the day because they've been exposed to small amounts of gluten proteins, right? So these are severe allergies, and shellfish allergies and peanut allergies are among the worst, right? There have been stories where children have eaten one small peanut or peanut oil in their curry or other type of like Thai food or something, and they die within an hour. Okay, so these allergies are severe, and a lot of physicians or a lot of research has been pointing to um, changes in gut microflora, changes in the cleanliness of our environment now in a Western society, where you don't see as many of these allergies elsewhere, in underdeveloped countries, let's say. So, autoimmune disease as well. So, it's an autoimmune disease is an immunological disorder in which the immune system improperly attacks its body's own molecules. So you see this with rheumatoid arthritis. You see this with lupus, right? So these are all autoimmune disease. You can also have Crohn's disease or ulcerative colitis as well, can be considered an autoimmune. Immune deficiency. So this is immunological disorder in which the body lacks one or more components of the immune system, making a person susceptible to infectious agents that would not ordinarily cause problem. Okay, so HIV causes immune deficiency, right? We talked about CD4 positive T cells, and there are a lot of HIV cases still in Africa, right? And southern Asia and the Pacific, right? And there's areas in Africa where the disparity is so high, and it's incredibly sad, right? Especially in Western medicine, where we've developed essentially drugs or medications that can prolong the life of individuals indefinitely, right? Even up to normal life, what we consider normal life standards, right? And in these areas, it's so endemic and so prevalent, and people are still dying from this disease. So it's not something that has gone away. It's gone away for us in North America because it's not a death sentence anymore. But other countries and other places in the world, it's still very much a death sentence to have HIV. Again, in these impoverished areas, in these impoverished countries, it's quite sad. So keep that in mind. HIV has not gone away. Just because Gilead or other types of biotech companies have created medicines and advances in the field that prolong the life of Americans doesn't mean everyone has the same access to the same resources as we do. So yeah. So pill that prevents HIV is no longer an abstract concept; it's a reality, right? And this pill again is incredibly expensive, and you've probably seen the commercials of this. They market it as kind of a prophylactic treatment to HIV. And this initially was one of the treatment strategies they would give with HIV in a cocktail, right? This actually is a faulty nucleotide analog, which means that it actually prevents HIV from elongating its chromosome or elongating its genetic information, essentially stopping HIV proteins from working appropriately. If you have this in your system already dissolved, and you get exposed to HIV, the virus has the inability to replicate inside of your cells because of the presence of this drug. Okay, so it's kind of a new, safer way, I guess, for certain groups of people that do have high numbers of HIV to protect themselves, and it's been quite amazing, the advent of new antiviral medications. I think I need to upload this PowerPoint, so I apologize. I haven't uploaded it. Okay. All right. So a couple of things before we get started, or before we leave. Sorry. You have a couple labs to do today. I'll be uploading the questions later. But you have blood typing, so you're going to be typing blood. Okay. So we kind of went over blood. So read through. This is going to be important for when we talked about a little bit about Mendelian genetics, and we talked about codominance, right? So you have codominance of the A and the B antigen, right? So O is recessive to A and B, but this codominance again will show through A and B, right? And essentially what you're going to do is the same blood test that you would do in a clinical lab, right? You're going to look for antigens. Okay, you're going to look for the antigens on the B and the antigens on the A, right? So the antigen is going to be, think of it as the what is present on the red cell. Okay, so think of the red blood cell. You have either the A or B antigen. The O is the absence of antigen. Okay, so what you're going to do is you're going to have these antibodies here. They're anti-A, anti-B. Think of this, right? This is an antibody that binds to A antigen. This binds to the A blood, A antigen on A blood, right? This is antibodies that are target those specific receptors on the red blood cell, okay, those markers, right? So antibodies that bind to A, what's going to happen? They're going to clump together. So if you remember the antibody antigen complex, so you see here, this clumping event, this is what essentially is going to happen when you expose the blood cells to the antibodies, okay? So this will only clump with A blood, this will only clump with B blood, okay? And then anti-D is going to be the rhesus monkey factor or the RH factor, right? RH negative, RH positive. If you're negative, you don't have clumping. If you add this and you see the agglutination of the clumping, see here the clumping, this will be a positive, right? So this person is A blood, this person, we'll take a look. This person is AB, right? B right here. And then this person is RH, whatever. So you have O and then RH negative or positive, okay? So pretty simple. And then I'll ask you questions about that later on here. And then one more. So here we'll have digestive enzymes. So we're going to talk about, I know we went through the GI tract, so we're going to talk about pepsin and different digestion of sugars, right? So we talked about the duodenum being innervated by different molecules. We talked about Benedict's reagent already, amylase. So you're going to go run through this procedure and see what types of enzymes dissolve what, okay? So Benedict's reagent is important for identifying sugars, okay? So being able to break amylase down into simple sugars. And you're going to find out what sugars those are, okay? So just run through the experiment, pretty self-explanatory. Okay, what else? I'm going to be uploading a slot for your paper so you can, actually your presentation rather, so you can so I'm going to show you this on my micro class just because I haven't done your project yet. Okay, so I'll upload it in week 13. It'll be scheduled for presentations, right? So you're going to come here and you're going to look at schedule for presentations. It'll be like this. It's not going to be able to access the window. You're going to have to click on the link. Okay, this link will bring you to a page, right? This Excel page. And what you're going to be doing is writing down your name under a date and time, right? And these are going to be the different times. You have a different lab times, but essentially these dates are going to be very similar to the dates that you're going to be presenting, okay? And you're just going to write your name down and a time that you want, all right? So it'll be during your lab time, actually for both lab times. So for both classes, I'll have multiple days that are going to be available, and you just pick one that you're able to show up. And if you're able to show up, write your name down, take it. If there are no lab times that are available that you can make, just let me know and we can schedule a time for you to present to me for a different time. So I'm going to have it in 15-minute slots. Again, your presentation should be 10 minutes. And I'll have like a 2-minute Q&A, and then that'll give me enough time for the next person to check in and start. Okay? Any questions about this? No? Okay. Oops. All right, so I think that's it for today. I think we're running a little late or a little long. Okay. Yeah, that's it for today. I think we are, yeah, that's gonna be it for today. I'll just go over the paper next time. I feel like I've talked your ear off. How long has it been? Over an hour. Yeah, we'll stop today. Right there. I'll go over the paper Monday, and we'll talk specifically about my expectations there. Okay. Any questions? No. Okay.